My name is Bohne. I'm a principal investigator in the Institute of Virology here at Hannover Medical School. My research is on gene expression of viruses and of cellular genes. So I'm covering both sides of the viral side, which tends to program the cell for its own production, and I'm also interested in cellular gene expression, which is also important in the patients to fight off viruses, but also to fulfill the normal function in a cell. My specialty are non-coding mutation. That means these are mutations in parts of the genome which are normally not translated into proteins. But this is the vast majority of sequences if you look at the whole genome. And the mutations are also more frequent in these regions. But it's harder to understand what they're really doing. And now with this patient collective of children which suffer from a severe infection of respiratory syncytial virus, we want to look at what are the genetic determinants in these children which cause these severe disease. Some of these mutations we already found, a few, but some of these are in the non-coding part. So they affect a mechanism which we don't understand yet. So in this course of research, you learn something about the virus. Why is this important? And you learn something how the cell works. Why does the cell use this sequence to regulate another process? So you're always learning from both sides. The cluster offers a unique cohorts, or many cohorts of patients. We have primary immunodeficiencies together with the group in Freiburg. We have what I talked about, the children that suffer a severe course of the respiratory virus infection. And we have a cohort of elderly patients together with the Department of Dermatology. So all this together gives you a unique possibility to look at these patients. Because for me, as a molecular geneticist, the gold mine are really the patients, because the mutations, if you find the same mutations in a few patients, you know this sequence is important. There's got to be something about the sequence which in the vast majority of the 3 billion base pairs you would never find and think, okay, this is important, but the patients guide you to the sequences which play a role in some processes. Yeah, I think that the big data, so you can summarize it like that, so the many sequencing data that are accumulating, either from whole genomes of patients or from the transcriptomes, so what from these genomes is transcribed into RNAs, then translated into protein. So there are more and more data accumulating from these patients. So we now not only get the clinical parameters like 30 years ago, now we get also the genomic sequences, and we know what genes are transcribed, and we understand more and more what they're doing. So in the end, this will give us a much more complete picture of what is really going on between virus and host. And then in the long term, I would envision that this gives you also an idea where to target the virus. So now where is the virus vulnerable? Where are new targets for antivirals? This is also another important part of the research. So I was always fascinated by this molecular arms race between the virus and the cell. So the cell, of course, wants to defend itself from the virus and then evolves some defense mechanism, but the virus is counteracting that. So it has a small protein, a small trick that fools the cell, that inhibits some processes in the cell. And this molecular arms race is really interesting to look at and to understand what are the mechanisms behind this, really.